Good day everyone, it's Yiko from Equity Tutorial. Okay, from our last class, I believe we talked about carbohydrates, monosaccharides and disaccharides. Well, today we'll be continuing that class. We'll be continuing with polysaccharides, polysaccharides. Now, unlike monosaccharides, which I said are just simple sugars, then I said disaccharides are a combination of what? Saccharides are a combination of two monosaccharides. Now, polysaccharides are made up of repeating units of what? Of the simple sugar. They are repeating units. They are aggregates of the monomers. They are aggregate. Now, let's imagine we have something like this: CH2ON. For a polysaccharide, N can be a very large number. Now let's imagine we have there are different monosaccharides, polysaccharides. We can have starch. We can have glycogen, and we can have cellulose. Starch, glycogen, and what? Cellulose. Now we know starch is the major form which plants store their food in. Glycogen is the major form in which animals store their sugar in. Then we know that cellulose is mainly found in the bark bark of trees and plants. Now starch as a polymer and glycogen as a polymer. Now when polymers form, when a polysaccharide is forming, there is usually the elimination of what? A water molecule. For a polysaccharide to form, there is the elimination of a water molecule. Now let's say a polymer like a polysaccharide like starch and glycogen. Now starch and glycogen, they are formed by linkage. Their linkage are mainly the alpha D glycopyranose. Linkage are mainly formed by alpha D glycopyranose, while that of cellulose is mainly formed by the what? Beta D glycopyranose. Now note this difference between starch, glycogen, and what? Cellulose. Now remember I said for a polysaccharide, N can be a very large number. Now let's imagine we have glucose. Now let's imagine we have a complex chain of glucose joining together, C6H12O6. Now this is the formula for glucose. Now bear in mind that N can vary from 5,000 to what? 10,000. N can vary from 5,000 to what? 10,000. And polysaccharides, they are very important in nature. Like starch. Now we know starch. Animals store food in form of what? In form of starch. Is a major form which they store food. Plants store food in form of starch. Animals store food in form of glycogen. Now just know these are very important stuff to note on polysaccharides. On polysaccharides. Now under polysaccharides, remember we said starch, animals in form of starch. Plants store food in form of glycogen. Then the most important polysaccharide here is actually what? Cellulose. Cellulose is one of the most important polysaccharides. Now let's do a quick recap. I said polysaccharides are what? Polysaccharides are giant units. They are giant units of what? Monosaccharides. They form very large units. And I said a polysaccharide is formed by the elimination of a water molecule. Monosaccharide is formed by the elimination of a water molecule. Now remember I said that when monosaccharides are coming together, they come together by what? Glycosidic bonding. They come together by what? Glycosidic bond. So we have starch, glycogen, and what? Cellulose. So under what? Polysaccharides. As a very quick recap. Okay, now basically next we're going to be talking about proteins. Now, of course, they told us in our secondary school or primary school what are proteins. They told us proteins are what? Proteins are things we take in, and when we take in, they help rebuild worn out cells. They told us chief source of proteins are meat, fish, etc. Today we are going to be looking at protein on a wider prospect. Now, what are proteins? Proteins are a very important component of all living things. In fact, they are very important. Now, proteins occur in many forms. Now, proteins help substance. For example, now if we are looking at a cell now, proteins are very useful in helping active transport, substances moving in and out of a cell. Proteins are very useful in metabolic processes, in building worn-out tissues. Proteins are very useful in enzymes. In fact, enzymes are basically proteins. Now, proteins are made up of what? Aggregation of amino acids. Amino what? Proteins are made up of aggregation of what? Amino acids. Now, what is an amino acid? You can just simply say an amino acid is like the monomer of the macromolecule, which is the protein. Now, amino acids have the general formula. Amino acids have the general formula NH2 CHRO. This is the general formula for the words amino acids. This is the general formula for amino acids. They have the amino group and the carboxylic group. The amino group and the what? Carboxylic group. Now, this is the general formula for proteins. Now, arrow can be just hydrogen. Arrow can be just hydrogen. Arrow can be an alkyl group. Arrow can be an alkyl group. Arrow can be an aryl group. Arrow can differ. Now, for the simplest form of amino acid, which is glycine, the simplest form of amino acid, which is glycine, we have it to be now for the simplest form of amino acid, which is glycine, arrow is hydrogen. Arrow is hydrogen. Now, the name of an amino acid is determined by the what? The functional group arrow is. The name of amino acid is determined by the what? Functional group arrow is. Arrow is hydrogen is the simplest form, which is not glycine. And note, apart from glycine, all other amino acids, especially the major amino acids, they are what? They are optically active, meaning the carbon here is what? Chiral what? Center. Apart from this glycine, all other amino acids, apart from glycine, all other amino acids are what? Optically what? Active. Now we're going to be looking at types of protein and the structure in which protein form. Now we can have the primary structure. We can have the secondary structure. We can have the tertiary. And we can have the what? Quaternary structure. Now, the primary structure of proteins are just simply long chains of what? Amino acids. The primary structure of proteins are just simply what? Long chains of what? Amino acids. Meaning amino acids that what? Form what? Straight chains. That is what the primary structure what? Of amino acids. Now, the secondary structure of amino acids. Now, from the primary structure, from the straight chain primary structure, in the secondary structure, they tend to be in regions along the primary structure. The amino acid will tend to start folding. They tend to start folding up or forming a zigzag structure. Now, this is because of the what? Of the hydrogen bonding. Now, note in the general formula for amino acid, which I just stated, you can see that on the amino group here we have hydrogen. And we know that from hydrogen bonding, hydrogen tends to bond with electronegative elements such as oxygen, nitrogen, and fluorine. Now, the hydrogen here will form a bond. The hydrogen here will form a bond with the oxygen on the what? Carboxylic group. Now, the secondary structure, there can be two different types of amino acids under the secondary structure. Now, we can have the alpha helix. And we can have the beta pleated sheets. Now, from the alpha helix, the alpha helix is just when two or more two or more protein group, when they combine, when there's a combination from the on every fourth carbon, what this simply means is that if we have a chain of amino acids, the first carbon will combine with the fourth, the second carbon will combine with the sixth, the third carbon will combine with the seventh, the fourth carbon will combine with the eighth. That is, there's hydrogen bonding between every fourth carbon. This will result in the alpha helix structure of the protein. Now, under the secondary structure, which is the beta plated sheet. Now, the beta plated sheet is just like when the amino acid group are arranged, they are arranged in parallel format to each other. Then there is hydrogen bonding between each amino acid group. This will form the beta plated sheet of the amino of the protein. Now, the tertiary structure. Now, the tertiary structure is the basic structure of a protein. The tertiary structure is the basic structure of a protein. It's just combination of the secondary structure. It's the basic structure. It is what really gives us the three-dimensional knowledge of the protein, and it also gives us like what is the true function. How does the protein? How does it really look like? That is what the tertiary structure. And they are mainly stabilized by non-local interactions like hydrogen bonding, which I just stated from the secondary structure, hydrogen bonding. They can also have disulfide bond linkage. Disulfide bond linkage and other interactions. Now, next is the quaternary structure. Now, the quaternary structure is just like an aggregation of secondary proteins. They are just like very large components, very large aggregation. Just imagine you are dealing with polysaccharides now. Now let's just imagine that towards quaternary structures. They are just large aggregates of this amino acid to form the protein. That is the quaternary structure of protein. That is the quaternary structure. So basically, I said we have the primary structure, the secondary structure, the tertiary structure, and the quaternary structure of proteins. Now, proteins, as we know, they can exist in different isomers. First of all, from chemistry, we know we can have the D form and the L form. The D, which is the dextrorotatory, and the L, which is the lavi-rotatory. Now, remember we said that proteins or amino acids they exhibit chirality. Now, note most amino acids, we find most amino acids in the L format, meaning they mostly rotate plane polarized light to the left. Now, note carbohydrates or basically monosaccharides, they are mostly found in the D format, meaning they rotate plane polarized light to the right. That is for carbohydrates, while this is for the amino acids. Okay, so basically we have looked at proteins and polysaccharides. In our next class, we are going to be talking about lipids, fat and oil, and lipids.